So we're going to look at the properties of monomers and the types of reactions that bond monomers together to form polymers, but we're also going to look at hydrolysis reactions that break polymers down into smaller monomer units. So the first reaction we're going to talk about are dehydration synthesis reactions, and that's what AP is going to call it, but sometimes in the past I've heard them also call them condensation reactions. So during these reactions, you're going to have two molecules combined to form one larger molecule. So these are reactions that build larger molecules, so they're in anabolic reactions. And the one thing that you're going to look for is that we're always going to have the loss of a water molecule, because we're going to have the hydrogen from one of the molecules combined with the hydroxyl, the OH group, from the other, and we're always going to have water that is a product. So dehydration reactions remove water and we will get a larger molecule. So anabolic reactions with the loss of water. All of these reactions in living cells require a catalyst. And remember, catalysts in living cells are enzymes. Enzymes are proteins and they speed up a chemical reaction without being used in that process. So here we can see a dehydration synthesis reaction or a condensation reaction occurring. So we have substrate 1 and 2. They're going to join to the active site of that enzyme, and they're going to go, and that's going to catalyze the reaction. It's going to create a favorable microenvironment for those to create the new product, and that product is released from the enzyme, but you should also know that water is also a product. Here's the opposite, having a larger polymer broken down into smaller units. So hydrolysis. Hydro means water, lysis means break. So hydrolysis reactions add water and break a bond. So you can see the water is being added, and that's going to break apart the bond between 3 and 4. Now we see that 4 is a monomer, it's the smallest subunit, and that we now have a shorter polymer that's comprised of 1, 2, and 3. But this is hydrolysis reactions always add water, dehydration synthesis reactions always remove water. Regardless though, every chemical reaction requires an enzyme, and sometimes we call these enzymes that break polymers down into smaller pieces hydrolytic enzymes because they catalyze hydrolysis reactions. So you can see in step 1 the active site is free and available for the substrate. We see in step 2 that sucrose, which is a disaccharide, is binding to the active site of that enzyme, and you see water entering that reaction, and at that enzyme now catalyzes the reaction to create two monomers, a monosaccharide of glucose, a monosaccharide of fructose. And we see that that water molecule that was added, it is now the H of the water molecule is attached to fructose, the OH of the water molecule is attached to glucose. So water broke the bond between those two monomers. So let's just go through really quick and talk about monomers and polymers. The building blocks of any biological molecule we call a monomer. So simple sugars, monosaccharides, those are the smallest subunit of larger molecules. Polysaccharides are called are many monosaccharides bonded together. Those are things like starch. Also things, we'll talk about that in a later screencast. Lipids usually don't form a lot of polymers, but we do form triglycerides. So fatty acids are smaller subunits of triglycerides, and triglycerides are an important biological molecule. However, like phospholipids, those really aren't big, huge, like you don't get phospholipids all joining together. Amino acids are the monomers of larger polymers like that are proteins, and then nucleotides make up nucleic acids like RNA and DNA. So let's look at a couple of these reactions. Here we have sugar molecules, and we have the dehydration synthesis reaction joining two monosaccharides to create a disaccharide. and you see that water is being lost. Water is a product of this dehydration synthesis reaction. So the reaction at the top is an anabolic process. The hydrolysis reaction at the bottom, that's a catabolic process because we have a larger disaccharide and it's being broken apart into two smaller monosaccharide subunits. So water is added, water breaks the bond between those two monomers, and now we have two monosaccharides in the hydrolysis reaction. This is the same exact reaction. We see two monosaccharides being joined through a dehydration synthesis reaction or a condensation reaction to create the disaccharide maltose. Here we see the opposite. We see a hydrolysis reaction because water is on the reactant side. Water is being added, it's breaking the bond between the two monomers, and now we have two smaller subunits, two monosaccharides on the product side. Here, this is showing two amino acids. Notice everything in blue. On the reactant side, you see that OH group, and you see the H. This is going to go through a dehydration synthesis reaction to create a peptide bond. So this is a condensation reaction. So we can see the OH and the H group, those are coming together to create water. So water is lost. It is on the product side. And now we have those two amino acids that are joined together to create a dipeptide. What I want you to notice is on this, when we're talking about the two amino acids coming together, one side with the nitrogen, that's the amino side of the amino acid, and the other side in red, right over here, this is a carboxylic acid or a carboxyl group. So when you join amino acids together, the carboxyl end always joins to the amino end. And we can see this happening again. So we see a dehydration synthesis reaction forming now a tripeptide. And I know it's a tripeptide because I'm counting the alpha carbon, or the carbon that's attached to the R group. So there's 1, 2, 3 amino acids that are all joined together. An AP question might be something like how many water molecules are formed during the synthesis of a tripeptide from amino acid monomers. So, and what we can see here is this is amino acid monomer 1 joining to amino acid monomer 2, and we would create one water molecule through that peptide bond, but then we create a second water molecule through the bonding of monomer 2 to monomer 3. So to create a tripeptide, that would require two peptide bonds, and we would create two water molecules. We also know that, I'm not going to talk about this in a great amount of detail, but nucleic acids, those nucleotides are bonded together through dehydration synthesis reactions, and they're also broken apart by hydrolysis reactions as well. And those bonds are going to be between the sugar and the phosphate ends of nucleotides. So normally, AP does not consider lipids to be polymers. They just make very big molecules, but they don't join together usually. The only time when I really do see lipids going through dehydration synthesis reactions is the creation of triglycerides. So you can see the glycerol part of the molecule on the left. It's a 3-carbon molecule, and then you can see 3 fatty acid chains on the right. So to make a triglyceride, tri means 3, and that's 3 fatty acids, and the glyceride part stands for the glycerol molecule that all of those are connected to. So to create a triglyceride, 3 condensation reactions happen between the glycerol molecule and the fatty acids.